Hello everyone and welcome to the course of Comprehensive Molecular Diagnostics and Advanced Gene Expression Analysis. This is the week 7th problem solving discussion and I am Udiptya from IIT Madras. So coming to this week's content, it was based on label, uh, based on mostly on proteomics. So we talked about label based protein quantification technologies in the first two videos, and then went about with discussing some label free methods for protein quantification. Upon that, there were another video on next generation proteomics, and lastly the proteomic data analysis and bioinformatic tools. Yeah. So, coming to the topic on introduction to quantitative proteomics. So, quantitative proteomics involves the identification and quantification of proteins within biological samples to understand their significance in physiological processes. How it gets important is that the variations in proteome abundance within cells exert significant influence on numerous physiological processes. And aberrant protein expression often accompanies disease development. So this is quite important because when you develop new diagnostics, drugs, essentially you need to understand how these biomarkers are getting the biomarkers are how new biomarkers are getting are coming, and you in during COVID, during any disease, yeah, some biomarkers will be getting overexpressed or getting down regulated. So essentially you can monitor the pattern of those, and you can actually detect several diseases, yeah. So these variations are quite important. So coming to its application, it's very important as I told on biomarker discovery. So this protein abundance, how it changes depending on your physiological condition, you can connect it a lot with the biomarkers, and you can tell a lot about the physiological condition. Understanding cellular signaling pathways, yeah. So a lot of uncovered mechanisms can be understood by this protein, understanding the abundance and significance of these proteins. Drug discovery and development, personalized medicine. So personalized medicine is a quite a new topic, or maybe a topic of recent interest, yeah, where you design medicines accordingly, yeah, depending on the profile of a patient. Yeah. Investigation of post translational modification and comparative analysis in systems biology. Yeah. Coming to several label based protein quantification methods, there are several methods. Out of this, we discuss here three. First one is isotope coded affinity tag. So this is a in vitro isotopic labeling using chemical reagents. Yeah. So it contains three elements: the reactive group for labeling amino acids, yeah, and isotopically coded linker and affinity tag biody. So this originally used deuterium, later redesigned with 13C to address chromatographic issues. Then you have iTrack and TMT. So these are isobaric tags. iTrack enables comparison of up to 8 samples simultaneously, whereas TMT you can go up to 16 different isotopic levels. So both of these involve labeling at the peptide level for relative quantification, and can be widely used in disease marker screening and drug target identification. Then you have SILAC, which is a stable isotope labeling with amino acids in cell culture. So this is an in vivo metabolic labeling strategy which uses 13C and 15N labeled lysine and arginine. So this enables very high labeling efficiency up to 99% and high precision. This is particularly useful for studying protein turnover and post-translational modifications. Coming to label-free protein quantification methods, so label-free quantification determines relative protein abundance without chemical labeling agents. So making it suitable to any sample type, including clinical samples. So two approaches are there. One is spectral counting method, which compares the number of identified MX mass spectrometry spectra which correlate with protein abundance, and another one is ion intensity method, which measures the area under the chromatographic peaks, or MS1 signal intensity. Advantages: it eliminates variability introduced by chemical labeling, yeah, and it's cost effective compared to isotope labeling methods. Simplified sample preparation with fewer steps, as you don't need a labeling step, and no limit on the number of samples that can be analyzed. Coming to its application, it has been used in disease biomarker discovery, monitoring proteome changes in biological processes, and studying protein interaction networks. Coming to data acquisition methods in mass spectrometry, this has been divided into three parts. First part is targeted approach, in which you have selective or multiple reaction monitoring, which essentially isolates and fragments predefined precursor ions and monitors predefined product ions. Yeah. And then you have parallel reaction monitoring, which isolates predefined precursors but records all product ions. Then you have, under untargeted approach, you have data dependent acquisition, which automatically selects precursor ions based on intensity or other criteria. And then you have data independent acquisition, which systematically acquires spectra across entire mass range without precursor ion selection. Then you have SWAT MS, which essentially is sequential window acquisition of all theoretical mass spectra. So it's a variant of data independent acquisition that divides scanning range into intervals, typically 25 Daltons. And it's a comprehensive analysis with high accuracy and reproducibility. This is often referred to as next generation proteomics. So you last in some I think last week's videos, or maybe last to last, we learned about next generation sequencing. Yeah, similar to this is next generation proteomics. Yeah. So coming to proteomic strategies and applications, so there are several strategies that have been presented. Yeah, one is bottom up proteomics, in which it involves proteolytic digestion before pass spectro and information reconstructed from identified fragment peptides, most mature and widely used approach. Then you have top down proteomics, in which it characterizes intact proteins without digestion, provides complete protein sequence and PTM information, requires high resolution instruments, FTIR or Orbitrap. Then you have single cell proteomics, which analyzes proteins at individual cell level, enables understanding of cellular and heterogeneity. Uh, it needs key steps. It has some key steps. First is cell isolation, protein extraction, quantification, and followed by data analysis. It has been very important in cancer research, immunology, and development biology. So single cell proteomics has been something that has been quite a good interest for people around working on this area. Yeah. So some bioinformatic tools that has been mentioned for proteomic data analysis. Some pre-processing tools. One is OpenMS, which is an open source software for LC-MS data management and analysis. Then you have Proteo Wizard, which provides tools for access and conversion of proteomic data files, and MaxQuant, which is designed for analyzing large mass spectrometric datasets. So these are pre-processing tools. Coming into identification and quantification, we have Mascot, which is a standard for protein identification from sequence databases. We have Skyline for building and analyzing various quantitative methods. For functional annotation and visualization, we have David, a Panther, which characterizes proteins based on gene ontology terms. We have Reactome or KEGG, which maps proteins to biological pathways. We have Perseus, which is an interactive exploration of large data sets. And then you have Cytoscape, which is a visualization of protein-protein interactions. Yeah. Future directions in this field essentially has a lot of scope. People are integrating AI, machine learning, yeah, in this. You have multi-omics data analysis that also is coming, and as we talked about personalized proteomics, yeah, and then cloud-based collaborative platforms that are coming ahead. Okay, then we can discuss the questions. Yeah. So each question I'll give you around 5 minutes, then we can solve it. Which of the following statements best describes the primary goal of quantitative proteomics? Yeah. So the answer is B. To quantify, quantitative proteomics is a field of study that involves the identification and quantification of proteins within a biological sample. Yeah. So, quantitative proteomics is widely utilized in biomarker discovery, understanding cellular signaling pathways, and drug discovery, development, personalized medicine, investigation of post-translational modifications, and comparative analysis in systems biology. Which of the following is a limitation of the original ICART technology that was later improved? So the answer is the use of deuterium caused problems in peak separation during liquid chromatography. So deuterium was originally used to develop the ICAR tags. However, deuterium interacts with the stationary phase of the column, which led to problems in peak separation during liquid chromatography. This limitation was addressed by redesigning the tags to use 13C instead of deuterium. In SELEC methodology, which amino acids are most commonly used for labeling and why? So the answer is lysine and arginine. The most utilized isotopically labeled amino acids in SciLac experiments are 13C and 15 and labeled lysine and arginine. This is because trypsin, a commonly used proteolytic enzyme, specifically cleaves at the carboxyl side of lysine and arginine residues. The combination of SciLac labeling with lysine and arginine are tryptic and tryptic digestion allows for overall proteomic quantification, as each tryptic peptide except the C terminal peptide will contain at least one labeled amino acid, enabling accurate quantification. Which of the following correctly describes the two distinct approaches used in level free protein quantification? So the answer is spectral counting and ion intensity methods. The level 5 approaches can be divided into two distinct groups based on the method used for data extraction: spectral counting and ion intensity methods. Spectral counting methods quantify proteins by counting the number of peptides or spectra assigned to a given protein, which correlates with protein abundance. Ion intensity methods, on the other hand, measure quantitative values through extraction of the area of the precursor ions. Chromatic graphs. Yeah, these approaches allow for protein quantification without the need for isotopic or chemical labeling. Which data acquisition method in mass spectrometry systematically acquires spectra across the entire mass range without relying on precursor ion selection? So the answer is data independent acquisition. So data independent acquisition systematically acquired spectra across entire mass range without relying on precursor ion selection. It captures data for all ions in predefined isolation windows. In contrast, data dependent acquisition automatically selects precursor ions based on intensity or other criteria, and only those selected ions are subjected to fragmentation in analysis. SR and MRM targeted approaches that isolate and fragment predefined precursor ions and monitor predefined product ions derived from the precursor. What is SWAT MS and what is its significance in proteomics? The answer is B, a variant of data independent acquisition that divides the scanning range into series of intervals for comprehensive analysis. It divides the scanning range into series of intervals, typically 25 daltons each, and obtains all information of all ion or fragments within the scanning range through ultra speed scanning. What is the main difference between bottom up and top down proteomics approaches? So the answer is B. Bottom up involves proteolytic digestion before mass spectrometry, while top down characterizes intact proteins. In bottom up, information about the constituent proteins is reconstructed from individually identified fragment peptides released through proteolysis. Top-down proteomics analyzes intact proteins, typically ionized by electrospray ionization, and analyzed in mass spectrometers capable of high resolution and accurate mass measurements such as Fourier transform ion cyclotron resonance or Orbitrap instruments. Which software tool is specifically designed for analysis of large mass spectrometric data sets with a focus on high resolution MS data and supports various labeling techniques as well as label free quantification? The answer is max quant. Sorry for the typo. MaxQuant is a quantitative proteomics software package specifically designed for analyzing large mass spectrometric datasets with a focus on high resolution MS data. It supports several labeling techniques as well as label free quantification. Which of the following tools is not primarily used for functional annotation of proteins identified in proteomic studies? So the answer is Skyline. Skyline is not primarily a functional annotation tool, but rather a software application for building selected reaction monitoring, multiple reaction monitoring, parallel reaction monitoring, and data independent acquisition methods, and for analyzing the resulting mass spectrometer data. The ICART chemical probe consists of three main elements. Which of the following is not one of these elements? The ICUT, so the answer is fluorescent dye for visualization. ICUT chemical probes consist of three elements: a reactive group for labeling an amino acid chain, example iodoacidamide to modify cysteine residues, an isotopically coded linker, and a tag for affinity isolation of labeled proteins. Yeah. A fluorescent dye for visualization is not a component of ICUT probes. ICUT relies on mass spectrometry for detection and quantification of labeled peptides and not on fluorescence. So this is a big question. A swath MS is being designed to analyze proteins in the mass range from 400 to 1200 Daltons. Calculate if each swath window is 25 Dalton wide, how many windows will be required to cover the entire mass range? If each window requires a scanning time of 100 millisecond, what is the total cycle time to scan the entire mass range once? If a typical chromatographic peak has 8 to 15 seconds, how many complete swath cycles can be occurred during the evaluation of this peak? And the last question, if the experiment is modified to use variable window widths that are narrower in dense regions of the spectrum with the window widths ranging from 10 Dalton to 30 Dalton, average 20 Dalton, approximately how many windows will be needed and what would be the new cycle time? So you can take around 10 minutes to do this whole question and then we will discuss it. Okay, let's solve it then. Then the number of windows, yeah, first question was on number of windows. So it will be equal to upper limit minus lower limit divided by window size, yeah. So it will be 1200 minus 400 divided by 25, which is essentially 800 by 25 or 32 windows, yeah. Coming to total cycle time to scan the entire mass range once, so basically it will be total cycle time will be equal to number of windows into time per window, yeah. So it will be 32 as we calculated into 100 millisecond. It is given. So it will be 3200 milliseconds or 3.2 seconds. Number of cycles, number of complete swath cycles during a 15-second chromatographic peak. Yeah. So number of cycles is peak width divided by cycle time. So peak width is given 15-second window. 15 divided by 3.2. 4.69 cycles are 4 complete cycles. And if the window size is changed to 20, averaging 20, so number of windows will be similarly 1200, or it will be from the first 800 divided by 20, which is 40, and time will be, cycle time will be 40 into 100 milliseconds, which is 4 seconds. Yeah. Okay. In a Silek experiment comparing protein expression between normal and cancer cells, a researcher labeled normal cells with light amino acids and cancer cells with heavy amino acids. After mass spectrometry analysis, the following data was obtained. So you have to calculate the following 3 things. So the first one, yeah, the fold change in expression for each protein in cancer cells compared to normal cells is protein A, it's 2.5 fold increase, yeah. So and protein B, it's 0.4 fold expression in cancer cells, or you can say 2.5 fold decrease, yeah. And then protein C, it's 1.1 fold increase, yeah. And coming to coefficient of variation, so for protein A, it's standard deviation. So how we calculate standard deviation? By mean, yeah. So it was like CV equal to standard deviation by mean. So for protein A, CV equal to 0.3 by 2.5 into 100%, which comes to 12%. Protein B CV is 0.1 by 0.4 to 100%, which comes to 25%, and protein C comes to 0.2 by 1.1%, which comes to 18.2%. The third probability that all sites will be labeled in a single molecule is, so for a protein with protein A with 5 lysine and 8, so it was written that 5, it's written that protein A has 5 lysine and 8 arginine here, and we have leveling efficiency of 98%. So 5 lysine, 8 arginine, and 98% labeling efficiency. So probability that all sites being labeled is 0.98 because of the percentage, and 5 plus 8, which is 0.98, which is 76.769, or you can say 76.9%.